Ladies and gentlemen, it's now my pleasure to announce the Charles David Marsden Lecture, which was created to recognize and to commemorate our second funder of the Society. It is usually given for contributions which have resulted in better understanding of the neurobiology of movement disorders. This year's lecturer, John Hardy, is perfectly fulfilling these criteria. He is originally coming from Leeds, and he trained at the Imperial College, went to the US, and finally became the chief of the Laboratory of Neurogenetics at the NIH, and returned to London in 2007. He has made major contributions to the understanding both of Alzheimer's disease, the genetics of Alzheimer's disease, Pick's disease, and Parkinson's disease. We are really pleased to have you here today, John.
Thanks. Really nice to be here. No. It's really nice to be here in Dublin. It's really a great honour to accept the Marsden lecture. Actually, the last grant application that I put in England in 1991 was a centre grant application with David Marsden, which was not funded. So, of course, it's well known that David Marsden and I share a similar dress sense. You know, it's really nice to take this award lecture. It's really nice, and it's actually especially nice to share the stage with my good friend and fellow Lancashire lad, Andrew Lees, who actually was responsible for getting me to come back to England by various manoeuvres, which I won't go into now. So it's really nice to give this lecture. It's a great time to be a geneticist, as you heard from the last lecture, which I thought was stunning. It's really a good time to be a geneticist, because if a disease is genetic, we can solve it. We don't need to be smart anymore. That isn't to say that the last lecturer wasn't smart, not at all. You just need to know which samples to collect, how to collect them, and have to be able to persuade grant funders to give you the money to do the work. That's all you need, because in fact genetics now is recipe driven. As you saw from the homozygosity mapping, we can now really rapidly approach genetic disorders. I think we're now at the stage that we can start to really be ambitious in what we ask, and we can say what causes Parkinson's disease. We can really start to get at that. However, I want to start by making a point that I think that these two papers represent perhaps the most important findings in the basic science of Parkinson's disease in the last 15 years. Showing this templating of the disease processes into graphs. We heard really nice lectures from Virginia Lee in particular, either yesterday or the day before, on how this is being exploited in terms of understanding the biology of the disease. And I think that we have not, as a community, yet absorbed the impact of this to think about what it means for the causation of the disease. And I'll come back to this at the end. I think it has profound implications, not only for the basic biology of the disease, but for how we might be able to actually determine how much of the cause of the disease we'll be able to find. And I think it has implications we have not yet considered. I'll come back to that with my last couple of slides. I also don't want to overstate genetics. I think that we have to be aware that genetics is not fate, and that identical twins are well known to be discordant, and indeed this is why, of course, the epidemiologists did not. I'm going to say get it right. No one can question me at the end of this lecture, so I'm allowed to be a bit ruder than usual. I think this is why epidemiologists possibly miss the genetic contribution to disease, because we can see that it really is not always concordant. Andy has identified a pair of identical twins which are nearly 20 years discordant, which have LRRK2 mutations, the pathogenic LRRK2 mutations, for example. And I think that this is something that we also need to be thinking about in terms of the causation. So we've been facing an issue which has been argued about, actually, and there's been several versions of this argument. In fact, we had this argument at Queen Square too. I have taken the view that we should define the disease pathologically, and my colleague Nick Wood, with whom I share a grant and a department, has taken the view that we should define it clinically. I think that we are both beginning to think that that perhaps that we should there is a middle line which we had not suspected, and I'll also come to that. I do think, though, it is important to think about the pathology of the disease. Now, with that background, and cognizant that Matt Farah spoke to some extent about this a couple of days ago, I'll go through the loci very quickly. I'll make a general point, though, and that is in most diseases, those people who have early onset have a more severe clinical course. And that's true in most diseases. It's true, for example, in Alzheimer's disease, where the presenilin mutations have a really severe clinical course. And that is not generally true in PD, and I think that is telling us something, that the course is different. And that fits with the pathology point I'm going to make. So let's go through them. I'll be as quick as I can on this. Part 1 is synuclein, described by Bob Nussbaum. Obviously, Lewy bodies. Maria Spallanzini showed Lewy bodies with the pathology of Parkinson's disease. Nasty, rather unpleasant disease, sometimes presents with dementia and often develops through to dementia. Part 2, very different. This is the first of those early onset disease, Parkin, described by Mitsuno. Despite its early onset, has a benign course, usually does not have Lewy bodies. I say usually, there are a couple of cases that have been reported with Lewy bodies, but each of those cases is rather complicated. Part 6, I have to say I would have predicted, this described by Nick Wood, has a very similar clinical course to Parkin. I would have predicted that this would not have Lewy bodies, but a very convincing paper really only one case, but a very convincing case described by Pau Pasteur with Lewy bodies. That was a surprise. It's a benign form of disease, however. DJ1, described by Peter Hertink, again early onset, benign course, no pathology reported yet. LRRK2, described by Tom Gasser and Matt Farah and by Andy Singleton and Nick Wood, very common cause of Parkinson's disease, have been lectures about that, usually has Lewy bodies, sometimes doesn't, and that was discussed by Matt a couple of days ago. Very common, surprisingly common disorder, relatively benign course. Now ATP13A2, early onset, very unpleasant disease, now known also to be a lysosomal storage disorder. Jose Bras and Rita Guerrero recently found it to be a lysosomal storage disease gene also. And this is a very unpleasant form of disease, starts in the late teens, death usually after dementia by the age of 30. PLA2G6 described, this has really had a nomenclature nightmare, but it could be described as Halle-Worden-Spatz disease, could be described as it was an INAD disease, and then with the nightmarish neurodegeneration with brain iron classification, sometimes described as neurodegeneration with brain iron type 2 without brain iron. That is a nomenclature committee gone crazy. And then Part 15 described in Iran by Elahi, early onset recessive disease. This is like a more unpleasant version of Parkinson's disease, no pathology. And then BPS 35 described, seems to be rather similar. We have one family described first by Matt Fowler and Alexander Zimpricht by exome sequencing, seems to be rather similar to LRRK2. So this is a large list of genes. We had a couple of other announcements from Matt a couple of days ago. I'm not convinced by his data on EIF 4G something, I can't remember the name, but I was not convinced by that, but let's digest them for a while and see if they turn out to be right. We heard a discussion about whether this part classification is good or bad. We had a similar classification discussion about scars, whether they should be called scars or not. I just think that this is unstoppable. I mean, we can say it's not good, but it's not going to change. It's bigger than movement disorders. People just, OMIM is bigger than movement disorders. It's going to be very difficult to persuade people to change this classification, and I agree that it's not ideal. But actually, no classification is ideal. I want us to think, though, that these are the foods in the fridge, and we've now got to cook a meal out of them. What pathways do they map out? Another member of this pathway, as we're going to hear tomorrow, is from Ellen Sadransky, is glucocerebrosidase. I put this just to remind me to say, of course, we understand glucocerebrosidase role in Gaucher's disease. Her work, which I'm sure all of you know, really shows that GBA mutations increase risk of Parkinson's disease by about 5-fold. I should just say that we have one wrinkle on this, in that Raquel Duran, looking largely in Tom Faulkner's series of cases, has found a mutation, E326K, which does not cause Gaucher's disease, and yet does predispose to early onset Parkinson's disease. So the relationship between Gaucher's disease and PD is a little bit complicated, undoubtedly, absolutely undoubtedly real, and this also, though, like what I said about ATP13A2, focuses at least some of our attention on the lysosome. Now, are we looking for one pathway? Are we looking to make this into a meal with one course? Are we looking to make this into a meal with 2 or 3 courses? Are we trying to find one pathway to disease, or are we trying to find several? Now, I think we know very well now, there's been some beautiful work starting in Drosophila with these 2 papers on pink and Parkin, also some very nice work from Mark Cookson on DJ1, showing clearly that mitochondria are there is one pathway, that one pathway at least involves mitochondria. And some very nice work which my colleague Helen Plum-Favreau has under second review at the moment, has also implicated FBOX07 in that pathway. And I'll just show you that pathway as far as we understand it now. And this is Helen Plum-Favreau's adaptation from Palank's Cell Biol review, showing how at least 3 of the genes interact in Parkinson's disease. Here on the membrane, you have pink, which is known to be a mitochondrial kinase. It's constitutively cleaved by an enzyme called PAL, so the concentration does not build up. When you get a mitochondrial damage, for some reason we don't understand, that constitutive cleavage is switched off. The concentration of PINK1 builds up in the membrane. It presumably phosphorylates something. It is a kinase. We don't know what it is. There is some data from the Dundee group suggesting it might indeed be Parkin, but we don't actually have that confirmed yet. But something happens when pink concentration builds up. And FBOX07 is brought from the cytoplasm to the membrane, as is Parkin, and Parkin and FBOX07 interact at the membrane. They're both ubiquitin ligases, and mitofusin is then ubiquitinated, and that mitochondria is then scheduled for mitophagy. So this is a way, it's the way of doing quality control on mitochondria. And if you're not doing quality control on your mitochondria, presumably your mitochondria concentration build up, and that starts to cause problems. Why that would cause problems so selectively in the nigra is not clear, though why it should be a very selective lesion. Remember that I said that this is a disease which does not really, it has a benign course. I'm sure many of you have patients. It has a benign course and remains relatively selective to the nigra. Why that selection should be, I don't know, of course, but of course it does. The dopamine metabolism is one possible reason. This is a difficult metabolic synthesis and possibly subjects the nigral cells to more damage. And I should say that there is nothing new under the sun, and this is work from Tony Shapiro and David Marsden and Peter. A list of famous people there. I hadn't appreciated the name of the institution, the Royal Kings and Queens Parkinson's Disease Research Group. Sounds really grand. We should reform that group. It's a really nice name. Anyway, there's been a mitochondrial view of Parkinson's disease based on the pathology for a long time, and so this fits with that view for sure. What about the other genes? For the other genes, we are in a lesser, we don't have quite as elegant data, but in fact they are beginning to show some signs of mapping to a pathway. VPS35 is involved in vesicle recycling from the endosomes and therefore in lysosome synthesis. GBA, of course, is in the lysosomes. LRRK2 seems to have some role through the mTOR pathway in controlling this fusion pathway, and so, what we do know about synuclein is it's involved, it is absolutely involved in vesicle fusion. So we can actually suggest that all of these genes are in fact involved in autophagy. And that really allows us to be perhaps a little bit, pull up a bit of a compromise about whether they're involved in one pathway or more, because mitophagy is a special case of, mitophagy, which I described on the right-hand side, is actually a special case of autophagy. And the nice thing about drawing the diagram this way is that it perhaps suggests why the genes on the left have a wider pathology than the genes on the right, where the problem is simply in mitophagy. The genes on the left lead to perhaps a problem in autophagy. Of which mitophagy is a special case. So perhaps we can start to compromise between the view that pathology is king and clinic is king by saying they're both kings and queens. Before I talked about the GWAS data, I just wanted to measure, to re-mention this kindred, which I'm sure you've heard of before, which was first ascertained by Manfred Munter and has Parkinson's disease with the age of 35. This kindred was collected by Manfred Munter and Katrina Gwynn and was mapped to this showed this very large kindred, which we were able to show co-inheritance of the synuclein gene. We couldn't find mutations in the gene until Andy Singleton found that what was causing the disease was actually a triplication of the locus. And since you've just had a 2011 picture of an Illumina array, which is here on the top right. This is one of the first Illumina arrays, so it's the same data but dating from about 2008, so it's not as nice. But we can see here an increase in density. Here in the fish, though, on chromosome 4, we can see a tandem array of 3 copies of chromosome 4. of the chromosome 4 synuclein gene. So what this is telling us is that expression is important. It's not just the quality, the sequence of the protein, but it's how much you make that is important. And that's something that really, I think, has informed our view as to how we should start to interpret the genome-wide association studies. Now, genome-wide association studies, these are the ways we are going to find low-risk alleles which are common in the population. What we're finding here is things in the population which will, which have a frequency of between, let's say, about 20%, between 5 and 30%, which increase our risk of getting, in this case, Parkinson's disease by from 1.1 to 2-fold. And this is simply all about numbers and collaboration. It's simply all about that. And the nice thing about Parkinson's disease is that of all the neurodegenerative diseases, the Parkinson's disease geneticists are, I'm going to say, the nicest. Since there's others that are okay, but the Parkinson's disease genetics community plays together very well. This is our consortium at the top, with the PIs of each component of our consortium. Nick Erickson from 23andMe, since the Google founder found out he had a LRRK2 mutation, 23andMe is, if you like, a subsidiary of Google, and they have really gone to town on Parkinson's disease, and they have collected many samples through their website, so we have access to their data. And then the other consortium, Tatiana Faroud, Heide Payami, Jeff Vance, and Rick Myers, have also collected many samples and done many analyses. And now what we have to do is start to build together the numbers. This is the thing that drives us. These are the power calculations. And let me just explain it briefly to you. Let's say you have something which has a frequency of the population of 15%. How many samples are you going to need to find that locus in an outbred population? Well, you simply read up here, you read up this line. And the number of samples you need is dependent simply on the odds ratio of the thing that you're going to find. So, for example, if it has a good thing to think about is ApoE4. ApoE4 in Alzheimer's disease has an odds ratio of about 3. That means that you only need to have samples right down here at the bottom of about 300 or 400 to find it. But you can see as you read up here that by the time you get to an odds ratio of 1.1, you're going to need 15,000 samples. And it's really a simple relationship, if you have your samples well matched between numbers and what you'll find. And this is the history of that work here very quickly. These were the first successful reports. We and others had unsuccessful reports which were underpowered. And these are the genes which came out, synuclein and tau. I'm going to talk actually a little bit more about both synuclein and tau. Then HLA found by Heide Payami in a similar size sample. I have to say that the pathologists, particularly Pat McGeer, had said HLA was important in Parkinson's disease in the early 1990s, so this really vindicates what he had said at that time. And then, as we started to get more and more, we started to find more and more genes. So all of the previous ones were confirmed, plus 8 new. This is published. I won't go into these 8, but it's published. And then this is the last thing, which was 11,000 Europeans, and now 13 new over these. So really a lot. This is in This is in italics because we did not do whole genome on these on the extra samples between here and here. We did a focused analysis. Where is this going? We've got two analyses planned. Meta 3, which is actually the genotyping is completed, and that involves all the people I showed in the slide a couple of goes ago, and we know that that's going to give us many more genes, and that's a genome-wide. And then the beautifully named Meta 4, which will just because it's the nicest name, will probably be the last one we do formally. And that, I think, will report early to mid 2013. And that you can see that what we're doing is we're finding more and more loci based upon simple sample numbers. This is what a genome-wide association study looks like. You've seen one of these already. This is from the study mid last year. And I'm just going to now talk about how we interpret these and how we try and go from these peaks, which are now multitudinous, to trying to understand what's going on. I'll just quickly mention the genes I've mentioned. Here is synuclein. This is the biggest hit. This is tau. This is the biggest hit in Europe. It's not a hit in Asia for a reason I'll talk about in a second. Here is LRRK2. In this analysis, it was just below threshold, but it's now quite a bit above threshold. This is normal variability in LRRK2 and is not mutations. And here is HLA, and these are other genes. But this has all been published. I'm now going to talk about a few of these peaks, starting with synuclein and tau, and then talk about how we're trying to go further than this. So we were aware, because of the work I've just described on the Iowa kindred, but also because of work we've been doing on PSP and tau, that we needed to have a method of understanding the genetic variability of gene expression. And what my group has spent, and Andy Singleton's group in parallel, Andy Singleton and Mark Cookson in parallel, have been doing is building up databases of gene expression. And we're close to releasing this data. We have done 150 brains, 10 brain regions. These are the brain regions. These are control brains, and we've both genotyped them and done whole genome expression in them. So we have a database where we can look at any SNP in the genome and say, how does that SNP influence gene expression? Andy and Mark have done a larger number of brains but a smaller number of brain regions, and we've designed the studies so that they're complementary and we can pool the data. So we actually have a database of 400 brains for some brain regions. Now let's go back to that synuclein hit. This is that synuclein peak on chromosome 4. What does it do? This is the hit here. Here's the synuclein gene. And what it does is increase the expression of synuclein. So those of us who have these SNPs, the SNPs which give risk here, have about, this is a logarithmic scale, but they have about a 10% increase in the risk in their expression of synuclein. So I had shown you that triplication of the synuclein gene gave you disease onset in the 30s. Matt Farah and others had shown that a duplication of the gene gives you disease onset in the 50s. Here I'm showing you that normal variability at the locus contributes to risk of disease by looking in this database. So in this case, it's a rather simple analysis. Now I just, though, want us to think a bit more about what we're actually finding. And this is a more complicated view of the synuclein locus and the risk at the synuclein locus. And it's in this paper here, which is published a couple of years ago. Here we have risk here, and here we have disease expression here. This is the Iowa kindred here. This is the duplication kindreds. These are high risk. They are rare, so they're colored in white. These are very rare. Then this is the mutation described by Bob Nussbaum and other similar mutations. These have normal levels of expression, but of course they have mutations, and so they also have high risk. What we have found in our GWAS is this dot here. We have found something that has a population frequency of, let's say, 15% in the population, and increases your risk of disease by about 10%. But that does not mean we have found all the risk of disease. What it implies very strongly is that there will be other people in the population, and this is going to be rarer, just like the distribution of height, who will express perhaps 25 or 30% more synuclein than average, and they will have a higher risk of disease than the ones that we have identified in the GWAS. Similarly, there will be people in the population who will have lower than average expression of synuclein, and they're going to have a lower than average risk of developing disease. So what I want you to be aware of is that the GWAS and the mutation seeking is indeed giving us some of the risk of the disease, of the disease at the synuclein locus, but what it is telling us is that there is more risk to find at that locus. So for each of the loci that we have found, we now have to dig into those loci and find out more risk at that locus. And so we actually can't, the estimates we have of how much risk we have found are underestimates. There's still risk to find. And what is true of synuclein is likely to be true of all the other loci as well. So there is more risk to find. And the nice thing, though, is that that risk is to some extent predictable. If 10% increase in expression increases your risk of getting disease 10%, then one would guess that 20% increase in expression is going to increase your risk about 20%. So we are on the way for finding more risk. It's not always so simple. I'm just going to talk about tau now, the tau hip, which of course was more surprising. I should say that this was first described in a candidate way by Larry Golby, but this is really showing this is the tau locus, and what I want you to, the tau protein is 6 isoforms, and I want you to look at this alternate splicing here. We looked now at the risk SNPs in tau to see what they did to tau expression. And I should say the tau locus is very, very unusual in the genome. It has 2 haplotypes. The interesting, the H1 haplotype, this was described first by Matt Baker when we were at Mayo. The H1 haplotype is found all over the world, and for example, in Asia, it is the only haplotype. H2 is found essentially only in Europe, where it is the minority haplotype and has a frequency of about 15%. This is an interesting polymorphism because this same polymorphism is found in orangutans, chimpanzees, and gorillas. So they are polymorphic in the same way as the human population is polymorphic. And the H2 haplotype has a totally different sequence to the H1 haplotype. It's separated from the H2 haplotype about 3 million years ago. And everybody, certainly every European with the H2 haplotype is fundamentally related to each other. It's really an interesting thing. This, it's described in these 2 papers, and the reason it is, and this slide was given to me by Rowan de Silva, the reason for it is that the haplotypes are inverted relative to each other. They're inverted on the chromosome. It's not possible to say which is the original version. As I said, all the other primates have both versions. The mouse, though, has the H2 haplotype. So it's an ancient and possibly frequently occurring inversion. What, though, is the effect on disease? It turns out that the PD is negatively associated with the H2 haplotype. Remember, I said that Asians don't have the H2 haplotype, and this is why the locus did not show up in the Japanese genome-wide association. The negatively associated haplotype H2 is not present in that population, and that's why it was not seen. What does it do? And actually, the only thing we have been able to find is that the H2 haplotype, the protective haplotype, is associated with more inclusion of exon 3. So more inclusion of this exon here. And that seems to be protective against Parkinson's disease. So it isn't a general effect on it is not a general effect on expression, but rather an effect on a specific effect on splicing. And this was partially suggested, I have to say, by Richard Wade Martin's group in Oxford using a cell biology approach. So but anyway, it's a splicing effect, not a general expression effect. This is, though many of you will know that there's an association with PSP and H1H2 too, that's a bit more complicated than this. I'm not going to go into this because this is a Parkinson haplotype, but there's something else going on. The H2 effect is part of the PSP effect, but there is another effect which we don't fully understand as well. I've given you two examples, synuclein and tau, where we are beginning to have some clear idea of what's going on. We know the gene, we think, and we know the effect. But this just shows you that there's still quite a bit to do. This is one of the other hits in the genome-wide association. And you can see this is the genes in this region. We can't even say which gene it is in this region yet, let alone what the effect on that gene is. And so there's still a lot of work to do to dissect this. There's still a lot of work to do. Now, how much risk have we found as geneticists? LRRK2 explains about 2% here in Northern Europe, much more in the Jewish and Arab populations. GBA about 5% here in Northern Europe, much more in the Jewish and Arab populations. Other Mendelian genes, let's say about 2%. Synuclein and tau, let's say the normal genetic variability. And actually, I should have put HLA there too, perhaps about 10%. So, and then the other GWAS loci, perhaps about 5%. So we're getting to be close to or over having understanding about 20% of the risk. And as I explained on that complicated synuclein slide, we have more risk still to find at all of these loci. So that number will go up. So as a geneticist, we have two aims. I've talked about this a little bit already, and I've talked about both, and I've shown how we have started to get pathways to pathogenesis. So we are beginning to understand the pathways to pathogenesis. Clearly, more work needs to be done. The statistical risk profiles, yes, we've found a reasonable amount, but we still can do and could do better. I don't know if we will ever get to be predictive in general. One thing I'd like to say, which I think we have not considered as a field for all of the neurodegenerative diseases, which I think we should offer patients more of, is pre-implantation diagnosis for some of these diseases. The amount of pre-implantation diagnosis which is being offered for nasty neurological diseases in general is absolutely tiny, and we could do a lot better and using the genetics in a way to directly help reduce the burden of disease. And we don't do perhaps enough of that. Let's think about how the field has changed. In 1996, this was the general view of where we were up to in Parkinson's disease and what the cause of Parkinson's disease was. I remember being on a study section when the Contersi kindred was discussed. I was not the reviewer, and the comments of the reviewers were that obviously the people in the kindred must share the same well water, that because it was known not to be a genetic disorder, and the grant was triaged. I remember that in 1996. So we have to be aware, you know, false knowledge is a dangerous thing, and those of you, you know, you should think about my lecture in the same way perhaps. False knowledge is definitely an inhibitor of progress. This is where we're up to now, I think. This is what we think about the disease now, genetics and environment. We're beginning to see things. But actually, I want to go back now to the first slide. And one thing we have not really taken on board, you know, perhaps we'll never find all of the risk. If a disease can start, those first papers I showed, showing that the disease can spread, imply that the disease can start with a single molecular event in a single cell. In other words, it can start stochastically, in the same way that we can think of cancer starting stochastically, or of course, prion disease starting stochastically. If a disease can start stochastically, that, let's say, by 6 molecules of synuclein bumping into each other in the wrong way, we cannot predict, we won't be able to predict it ever. And those identical twins who are discordant, maybe then it wasn't anything different in their environment. Actually, as far as we can tell, they have had very similar lives, but rather it could be something stochastic, and that we will never be able to fully understand it. The other thing that is very difficult to tie together, I think as I showed, we're making some progress with pathways. It's very difficult for us to tie what I said about synuclein spreading and so on. It's very difficult to tie that information into the genetics of the disease at the moment. So there's clearly big knowledge gaps. I think we have made big progress, but there's going to be plenty of future Marsden lectures, I hope, where other people will explain further the rest of the journey we clearly have to make. Thank you very much.